Okay, this is a demo of 7P. Um, Although you won't have much to do with the graphics part of it, you have to prepare the data that will be properly handled by the graphics. Um, so if you run as the interactive mode, there's a default file that's opened up, and that file and two others are in the starter code. Just to see how the layout is here, that the files that get opened have multiple contigs, which are sequences of DNA that have been determined to belong together and match the real organism. These are seldom full chromosomes, but depending on how good the assembly and the sequencing is, they can be pretty large sections of the DNA. The samples we have are pretty large, at least the larger ones. But I've cut them down into this particular, the initial one only has two sequences in the file. The original organism has several thousand. They are the longest two, so they are the closest things to a chromosome that this organism has, or at least had at the time I got the data. Once we get to this part of the display and what you've done your part of the code, we're seeing a single gene, which is a piece of a single gene from gene 1, and down here in the bottom it gives you the gene number. It's the length, well, yeah, this is from gene 1. The length of this CDS, which is the portion of the gene that codes for protein, coding sequence, CDS is coding sequence, is a little over 2,500 nucleotides. Each nucleotide is a molecule. The gene itself is composed of alternating exons and introns. Exons are part of the coding sequence. If we make this smaller so that we're not using as much space. So from this red T over here to this red T here is an exon. That is a piece of the gene that will be combined together to form a coding sequence, which eventually will be translated into, transcribed into a protein within the cell that will do something as part of the living process. From this point to this point, these sequence of nucleotides are in the gene. And then right here, after this T, there is another set of nucleotides that are not part of the coding sequence. And those are called introns. And then this intron is stuck right here between the red T and the blue C. And it doesn't matter really what those are right now, but it's actually stuck there in the gene and expanded out, and it's quite large. It's roughly the size of the exon, and then there's that region stuck right in that place there. And then that intron ends, and right here with another blue T starts a new exon. So that's exon 1 and then exon 2. And between the two is intron 1, or intron 0 probably. And then come along here, that exon ends, and another intron is sitting in there. These aren't overlapping in real life. All of this intron is squeezed between those two, and all of this intron is squeezed between those two. But in order to form a mRNA, which ultimately gets transcribed into a protein, these intron pieces have to be ignored, and the exons have to be concatenated together to make a coding sequence. And so that's what, we can make this as small as we can here, that's what we see here, and the purpose of this program is to show the coding sequence, which goes on for quite long, almost 2,600 nucleotides, and then where in that coding, within the gene, that coding sequence is interrupted by introns. This has 16 introns and 17 exons, and that's the normal thing. You'll start in the gene, you'll start with an exon, then an intron that breaks it up, and then another exon, intron, exon, intron. And so most of the time you'll have one more exon than intron, and then eventually you get to the end of the coding sequence, and then what follows is not part of the gene per se. I mean, there are a few more nucleotides at the beginning and end that we call part of the gene, but it isn't coding sequence. Okay, so your job is to find in the files the sequence information and the exon intron information so that you can prepare objects to be fed to this program so we can display them. And at any time, there are just a couple of options here. Here you can open a new GTF, gene transfer, can't remember what the F is, and sequence files that go together. All the information about where there are exon and introns are in the GTF file, and then the actual sequence data, the nucleotide information, is in the sequence. And so your job is to combine those two files and extract the necessary information so that you can provide the information to the drawing program. Right here, we can show the coding sequence in this, the information about this gene, which has the coding sequence here, by simply clicking on it. And here we have, and this is all information that your program needs to compile and produce in this form. Primarily you compile it. I'm providing code already that will ask your code for the information in a certain form and then format this. So here we have, this is gene 1, it's on the negative strand. If you had biology, the DNA is 2 strands, one going in one direction, one going in the other. They are complements of each other, so if you know one of the strands, you know the other. But when we sequence the data, we have no idea which pieces of the DNA have because the DNA, we only sequence one strand at a time, but it's each little piece is only one strand, and we don't know which is the forward and which is the backward, and that doesn't really matter for your program. Contig 0 is where it comes from. Phase, we just report, we don't use it. And this gene within the contig starts at position 4358 and goes to 9245. The gene has 17 introns and 17 exons and 16 introns, and the coding sequence starts at position 319 within the gene, and it's 2595. And then this is the information about the exons. Starts at position 0 of the coding sequence. It's 161 nucleotides long. The first 4 nucleotides are TCAA. The last 4 are GGAT, and there's 161 between them. And then these are the introns. We can get the same information. We'll show any gene, but that requires that we type in the gene ID. And you can give, oops, not G9, but G1. and you can give a number which says how many of the leading and trailing, the beginning and ending nucleotides do you want printed. So in this case, we'll do 10 instead of the default 4. And so now we get the same information, except that the program takes the first 10 nucleotides in the sequence and the last 10 just as a check if you wanted to check. The idea, there are copies of these as TIFF files in the starting code, just so that you can make sure that without having to actually count nucleotides in the DNA files, which is tedious, try to match, make sure you get something like this. There's a good chance you'll be one number off. The GTF file numbers positions beginning at 1, and of course in all of our code and text sequences and so forth, arrays, we begin at 0, so it's easy to be one count off, but that's also easy to fix once you notice it, if you know what is right. So at show any gene, we can also show G2, and I'll just show that because they are quite different. I'll do 20 this time. You don't need to do that. But so gene 2 is much shorter. It only has 480 nucleotides in the coding sequence. And it only has 4 exons and 3 introns. We'll open a different GTF and sequence file. All you have to do is open the GTF file, and it will automatically open the corresponding sequence file. Oh, my solution has many more files than I provided for you. Here's another one that is, so this is from Contig 1, and in Contig 1 there are a few genes. The gene numbers are unique across all contigs, so, but this is a much smaller one, and a much smaller gene. If we look at, coding sequence, but the same sort of information is available. And there's one other file that I've given you, which is got quite a few more things in it. I think it's this one. This has I don't remember. The ones I've given you, I've also given you snapshots of this portion of the view, getting as much of the DNA as possible into the screen, just so you have some clue that you're coming close. And I think that's all you need to know there. Now, there's one other useful thing. I'm going to show this. So there's here, this shows that one of the genes, gene 5, oops, gene 5, which is that one, actually has 7 exons and 9 introns. And that can happen when you find one of those. You really need to just ignore it. Indicate the fact that you found it, and what that means is there's an exon, that there's an intron that is past the end of the last exon, end of the coding sequence. There is portions of the DNA past the coding sequence that are important and can be interrupted by introns, but they're not of interest to us for this particular assignment. It just means you have to be careful that when you find an intron, it may be outside the coding sequence and you want to just ignore it. So even though it says there's 7 exons and 9 introns, and not all of them. The three introns that are past, in this case, they're past the end of the coding sequence. We don't even show here, but you need to record. You can also get introns that precede the start of the coding sequence. So those are just small little things you have to be careful about, that they don't greatly complicate the program at all. So the other thing is that in order to help your debugging and in your implementation and implement things a bit at a time, the GTF data file, which is one of the two that you class, one of the two things you'll be implementing are GTF data and gene.java. Has the GTF data has a main that does lots of the preliminary, invokes some of the very preliminary processing of the data, the reading of the data, and the initial distribution of creation of the exon intron information, but does not require that you get the sequences properly extracted and so forth to get pretty far along the way, and does not require the use of the graphics tool. So when you run this tool, and there's a default, and you can provide command line arguments, it will read the file, call mostly gene code, which has to build all this information, and then prints it out in a form without having to go through the graphics part. There's less if you have to use the debugger and set breakpoints, if you have to put in print statements and so forth. There's a whole lot less code you have to go through in order to get this far. And when you do have everything working, all of this information will be available, and it's the same information that then is then used, including extracting the DNA. But you can get this part working before this will show up correctly. You'll just it won't be there, because my code will call that displays this will call you for this information, and you'll return null, and so this just won't be here. But it's a tool that you really need to use to help debug your early stages of programming, so you don't have to implement everything before you start testing. These log files are also for the three data files I've given you. I have the log files that my program produces from running it through the GTF data main. And so this is the same one. This is from a little water snail called C. brenneri, and it's config 0, gene 1, and gene 2. Not that that matters, but that's how the logs are labeled. Oh, I forgot to mention too that for the I also have taken snapshots of this for each of the genes, I guess, and this page. So there's this isn't hundreds and hundreds of lines of code, but it's you have to be careful to read the specifications and ask questions about whatever part of that the handling of the strings. It's mostly a string handling problem, although there's also data manipulation. You've got to build a hash map to store the information about the exons and introns and where they are. It's not a lot of code, but don't wait for the last minute to work on it because you will have questions and need them answered. Okay.